Hey guys, welcome back from spring break. Hope you were able to enjoy some of your time, even if it wasn't exactly what you had planned. But I'm excited to be back with you guys and finishing out our Unit 6 on protein synthesis. And the last thing we need to talk about is gene regulation. Okay? Because, like we learned with the microarray lab, not all genes are turned on in all cells all the time because that would just be a huge waste of energy. Many of you gave great examples in your lab report telling me that like a muscle cell doesn't need the same proteins a skin cell needs or a stomach cell needs. So those genes that it doesn't need will be turned off, while the genes that it does need will be turned on. But here we're going to start answering the question, how do we go about turning genes on and turning genes off? And because we deal with two different major classes of cells, prokaryotes and eukaryotes, there's two different mechanisms that this occurs through. And as you could probably imagine, the prokaryotic method is a bit simpler than the eukaryotic method. So this activity that we're going to be working on with today's class time as well as our next class time, either Wednesday or Thursday, is going to focus mostly on the prokaryotic method of gene regulation. So let's just walk through this picture because today what you're going to be doing to start off this activity is you're going to be watching this video as well as an additional video on operons, which you'll understand that word shortly, and answering these pre-lab questions, of course not in your lab notebook, but on a Google document in your Google Drive folder. And please make sure it's clearly labeled so that I can find it. Okay, the second part of this activity is going to have you working through the Biograph simulation. Remember when we did those surveys to get us set up to do Biograph? Well, we're going to be working through our first simulation. The procedure is listed in the same lab packet. And then, of course, there are some analysis questions which you are going to be answering on the same document you answer the pre-lab questions by the end of your class period on either Wednesday or Thursday. I'm giving you everything up front in case your pre-lab questions don't take you that long today and you have a little bit more biology time to fill out. You can keep moving through the simulation. Remember, you're supposed to spend about an hour on biology for each class day. Okay, so if the pre-lab questions don't take you an hour, move on to the next part. All right, so let's talk about this picture quickly. So what we see here is a piece of nicely color-coded DNA, and there's several regions on the DNA. There's a promoter, which we've talked about. It's where transcription initiation happens. There's an operator, and then there's the coding region. This is the part that actually holds the instructions for building the protein. So when a gene is turned on, RNA polymerase can easily bind to the promoter, move past the operator, and go straight into the coding region building mRNA. RNA polymerase is able to find the promoter in a prokaryote thanks to the help of an activator protein. So as long as this activator protein is present and there's nothing blocking RNA polymerase from moving through to the coding region, transcription will occur. However, it's possible that another protein called a repressor could sit itself right on the operator. Now, if this happens when RNA polymerase binds to the promoter, it's not going to be able to move any further, and this coding region will never be transcribed. This gene is now successfully turned off. And in prokaryotes, this coding region usually holds the instructions for several related genes. So if it takes 3 different enzymes to break down a certain macromolecule, this coding region will contain the instructions for all 3 enzymes to be made at one time. That's what we refer to as an operon, which is something that's explained further in the next video and something that you're going to be exploring using the Biograph simulation. Eukaryotes don't have these operons. They have one promoter for each gene. But in a prokaryote, one promoter could control the transcription of multiple genes. Okay, pretty simple, straightforward. If there's no repressor on the operator, RNA polymerase will move right through, carrying on transcription. That mRNA will then move on to translation. In a eukaryote, a gene is typically turned off if it's pretty tightly wrapped around those histone proteins. Okay, because if a gene is wrapped tightly around histone proteins, RNA polymerase cannot get in to bind to it. So it's pretty successfully turned off. If the winding of this DNA strand loosens a bit, then RNA polymerase can potentially get in. But RNA polymerase is not going to work alone in a eukaryote, and it's not going to necessarily be as simple as just having one activator protein help it find the promoter. There are multiple different protein factors that help RNA polymerase bind to the promoter, and the promoter of a eukaryotic gene usually contains this sequence TATA, thymine, adenine, thymine, adenine. We call it a TATA box. So once all of these required activators, also referred to as transcription factors or enhancers, are bound to the DNA upstream of the promoter, meaning before the promoter, then RNA polymerase can bind to the promoter and move on to transcription. All of this protein binding can only happen if the DNA is not tightly wound around the histone proteins. All right, so with that information and some of your prior knowledge, you should be able to answer these 5 pre-lab questions in a document in your Google Drive folder. In the next video, I'm going to do a trial run of the simulation just so you could get a sense of what that is, and I'm also posting a video from YouTube that talks about an operon in a bit more detail. Today, what I'm also going to try to do is take a picture of your microarray lab report rubric and drop it in your Google Drive folder so you could see where points were lost, but overall they were very, very, very well done. I was very impressed, and it's making me excited and looking forward to our quarter 4 lab report, which will be your final project lab report. All right, guys, stay well. I'll be on Zoom at 1 o'clock if you have any questions. Talk to you soon.